We're dealing with multiple audiences in multiple presentation modes, so don't hesitate to yell out if there's some kind of error as we go. And also, we're getting started a little bit late, and I may be presenting material that is a little bit tangential to what is normally presented in this venue. So if we get through only a handful of slides, I'm perfectly fine with that. If this winds up being more of a discussion than a presentation, that would be great for me. So don't hesitate to stop me, ask questions, so on and so forth. Okay, so let me begin by stating the obvious, and that is that the value of our goals is dynamic and subject to change. Case in point, this now old picture of my son Benjamin, who is reaching for this highly rewarding board book. But he's getting an aversive consequence, this stiff arm to the face from his sister Isabel. You can see that that's evoking a negative affective response in Benjamin. And I can assure you that if this had been a video, the approach behavior that Benjamin's exhibiting here would have rapidly changed to avoidance behavior. And so I'm interested, my lab is interested in everything that you see going on here. We're interested in how the central nervous system and related signals and circuits encode affective information and convert that affective information into motor plans to either approach or avoid. But we also recognize that the value of any stimulus is not set in stone, it's subject to change. And it can change based on learning, like what you see going on here, or changes in our physiological state, for example, how hungry or sated we are. And hopefully that's a tie-in with the group here. But this sensory to motor conversion has served us quite well over the eons. It's what helps direct our behavior towards beneficial, nutritious, rewarding stimuli. Am I good?
I think so. Went black for a second.
Okay. Okay. Helps direct us to rewarding stimuli, helps steer us clear from harmful, poisonous, aversive stimuli. But more recently, it's gotten us into a little bit of trouble in that sometimes it steers our behavior towards nutritious, rewarding stimuli, but stimuli that perhaps is overly nutritious. And here's an example of a donut burger, which provides plenty of calories, so much so that we wind up taking in too many calories and we wind up storing that as fat, and all kinds of bad things happen from there on out. And in thinking about the central signals and circuits that underlie this sensory to motor conversion, some people have painted a very simplistic picture of circuits that are sensing energy in and control energy out. And I kind of think that these very simplistic renderings of the circuitry involved, the hypothalamus, the brainstem, kind of remind me of South Park's underpants gnomes. These are creatures that steal into the rooms of boys in the middle of the night and steal their underpants. And one night, one of the boys wakes up, confronts the gnome and says, "Why are you doing this?" And the gnome says, well, here's our three-part plan for why we steal underpants. Phase one, they collect underpants. Phase two, phase three, they profit. And of course, all of the action is in phase two, and it's relatively unknown. So what I like to do is dig into the phase two part of the sensing of caloric intake and the generation of behavior. But when you dig into phase two, it can get quite complex. And so this is a rendering from Berthoud et al. Very recently, 2020, trying to capture all of the signals and circuits that are involved in feeding behavior. My lab focuses in on just a small part of this puzzle here, the mesolimbic dopamine system, but we recognize, so I'm going to zoom in on that system, but we recognize that it sits within this much larger, very complex circuit. And before I get to the specifics of what we do, I just want to provide a quote from this paper here. The current obesogenic environment impinges mainly on a critical pathway linking hypothalamic areas with the motivational and reward systems to produce uncompensated hyperphagia. And so that's where I want to make my stand, where I want to delve in, where I want to investigate. And so to do this, we zoom in on the mesolimbic dopamine system. And in my lab, we try to record from that system in real time in behaving animals. And we do this in a number of different ways. We imbue the dopamine neurons with a fluorescent calcium indicator so that when dopamine neurons become active, intracellular levels of calcium rise and trigger this fluorescent calcium center, which fluoresces, and we can capture that fluorescence with a chronically implanted fiber optic. And just to give you a quick taste of what this might look like, you know, my lab, and I should just, some of my lab members are here, so I want to make it about me for a moment, and that I try to do, I'm a simple man, I have a simple mind, and I try to keep my science as simple as possible to make sense of it. And so we do very simple experiments. And one experiment that we do is we install an intraoral catheter subcutaneously so that I can deliver, or we can deliver, small amounts of fluid directly into the oral cavity of the rat. I just want to show you what all this looks like together in a movie here, where we simply just make a quick intraoral infusion of a sucrose solution, 0.3 molar sucrose, which rats like. And when I start the video, 5 seconds in, the infusion will occur, and then down below will be the dopamine signaling in the ventral tegmental area, time locked with the behavior that you see up above. Here goes. There's the infusion. It lasts just 5 seconds, and then the infusion is over. Hopefully what you can see is that as soon as the infusion starts, you start to see the animal licking. And at the same time, you get this massive spike in dopamine signaling in the ventral tegmental area. Playing the video again, but we also, in addition to recording the activity from the cell bodies of the dopamine neurons, we can actually record dopamine release in terminal regions like the nucleus accumbens. And again, I want to show you a video of what that looks like. Exact same situation here, brief infusion of sucrose into the animal's mouth. But here we're measuring dopamine release in the nucleus accumbens. Here it goes, 5 seconds in, the infusion begins. Start, big increase in dopamine release, and then the infusion's over and we get back down to baseline levels. So this is the foundation of the work that we do in my lab. Everything that we do is sort of built on this type of approach where we're measuring the mesolimbic system in real time in behaving animals and give our animals a variety of different taste stimuli to sample. And so today what I thought I'd do is address a couple of questions with you. First, I want to ask whether the mesolimbic dopamine system, which we colloquially know is involved in reward and reinforcement, ask if it differentially encodes rewarding versus aversive taste stimuli. Spoiler alert, the answer is going to be yes, but I'm going to show you a little bit of data to support that. I also want to ask whether this encoding by dopamine neurons is flexible and can change based on learning or changes in our physiological state, for example, hunger and satiety. And finally, I want to ask if the dopamine signaling is flexible, well, how do the dopamine neurons come to know about physiological state? How do the dopamine neurons know that the animal is hungry or sated? So I'm hoping to address those three questions with you today. We'll see how we do with time. So first things first, first of all, very simple question. Is there a difference in dopamine signaling in response to rewarding versus aversive taste stimuli? So once again, we make intraoral infusions of taste stimuli. And the nice thing about using intraoral infusions is that I can put whatever I want into the animal's mouth and it's obligated to sample or taste what we put in there. We can give rewarding stimuli, but we can also give aversive stimuli, and the animal is required or obligated to sample or taste whatever we put in there. So here in this old experiment from 2008, we just gave intraoral infusions of sucrose in green You can see the dopamine signal goes up. You've seen that in the movies already. But look at what happens if we give an aversive stimulus, quinine, which is bitter. You see the polar opposite dopamine response, a decrease in dopamine release. We've replicated that more recently with fiber photometry back at the dopamine cell bodies, and you see the exact same profile of results. Increase for rewarding sucrose, decrease for rewarding quinine. But we're still looking at apples versus oranges here, sucrose versus quinine. So we tried to come up with situations where we could give the exact same physical stimulus and have it be either rewarding or aversive for our animals. And to do that, we turn to a paradigm known as conditioned taste aversion. So it goes like this. We take naive animals, we make intraoral infusions of sucrose, which the animals like. And then as soon as that session is over, we give half the animals an injection of lithium chloride, which makes them ill. The other half get saline. Those animals that get the lithium chloride start to pair that illness with the taste of sucrose, which they had never had before. And they develop a conditioned taste aversion. The next time they get sucrose, they treat it as if it's highly aversive. Same stimulus, changing the value of the stimulus based on learning. We do this, we make 3 pairings. Let me just tell you the rest of the design because I think it's important. The next day in this design, the animals that got saline paired with sucrose get just an injection of lithium chloride. The animals that get sucrose paired with lithium chloride the next day get just an injection of saline. So every animal has had exposure to all the stimuli in this paradigm. Some have had the sucrose explicitly paired with lithium chloride and develop a conditioned taste aversion. Some animals have never had the sucrose paired with lithium chloride and never develop a conditioned taste aversion. We do this conditioning phase 2 more times and then test the animals in extinction. All the while we're measuring the dopamine response to sucrose in our rats. So now what I thought I'd do is I'd play the dopamine response to intraoral infusions of sucrose first in these unpaired animals that have never had the taste of sucrose paired with lithium chloride. So this is just the conditioning phase. You can see 2 times over, exact same dopamine response day after day. The value of sucrose isn't changing. It's rewarding each and every day here. What about in paired animals? Watch the change in the signal here. As the animals learn the relationship between sucrose and lithium chloride, it becomes aversive, and that dopamine response shifts from going positive to going negative. Then we test the animals in extinction, no more injections of lithium chloride, and you can see in the unpaired animals, sorry, just going to replay. In the unpaired animals, over extinction in blue, day after day after day, exact same signal. And it's so boring, I'm just going to switch on over to the interesting group, the paired animals. Watch what happens in extinction for these animals. As the animals come to learn that actually sucrose is safe, it's no longer making them ill, that dopamine response starts to migrate and starts to return back to pre-learning levels. The sort of naive levels. Take-home message here is that the dopamine response to the same stimulus is highly flexible and can change based on learning, like what you see going on here. Increases when the stimulus is rewarding, decreases when the stimulus is aversive. I just wanted to share a little bit about how we analyze these data. On the top right are the 2 distributions of dopamine responses during the intraoral infusion on the conditioning day for paired animals that will be paired versus animals that will be unpaired. They overlap perfectly. And if we do a receiver operating characteristic analysis, any line that you see that follows the line of equivalency means there's really no way of telling whether a single sample comes from one distribution or the other. But look at what happens after the animals learn the conditioned taste aversion. In red, we have the paired animals. In blue, we have the unpaired animals. And you can see now After conditioning, there's a big deviation from that line of equivalency. And here we're just measuring area under the curve, and we can statistically compare to the very first conditioning day when there's no difference, and then track this ROC curves over extinction. And you can see that eventually that difference between the distributions in unpaired and paired rats goes away over the course of extinction. We are interested in asking a lot of additional questions based on what I just shared with you. We're interested in the different excitatory and inhibitory sources of information to the ventral tegmental area, how those weights might change over the course of learning and extinction. We think we have some really good targets in the amygdala, in the bed nucleus of the stria terminalis, and others. And also, we also need to recognize that dopamine release might differ with respect to this paradigm in different terminal regions. So everything that I just showed you are from dopamine neurons projecting to a region of the nucleus accumbens called the lateral shell. And it could be that dopamine is doing different things in different dopamine terminal regions. So these are additional questions that we want to address. But I want to shift. We can come back to this if you have questions, but I want to shift to something that I think this group might find a little bit more intriguing. I just showed you how the value of a single stimulus can change as a function of learning. Now I want to talk about how the value of a stimulus can change as a function of physiological state. And I want to start with something that might seem a little bit odd, and that is the taste of concentrated salt solutions. If we're at the beach one day, we're in the water, we get a mouthful, beach, we're in Chicago. Some people talk about going to Lake Michigan as going to the beach. If you're at the ocean and you go into the ocean and you get a mouth of seawater, it's quite aversive, super salty, highly aversive. Same is true for rats and other animals, but animals in the wild often go through periods of low sodium in their diet. And when that happens, when they are deprived of sodium for a long period of time, they develop what's called the sodium appetite. And now they seek out concentrated salt substances so that they can replenish that needed sodium. And we can do this, we can replicate this in the lab by giving rats a diuretic that causes them to excrete large quantities of water and sodium, give them back water, but no sodium, a sodium deficient diet. 24 hours later, the animals treat these normally aversive concentrated salt solutions as if they're highly, highly rewarding. So again, we can measure the dopamine response to intraoral infusions of a concentrated salt solution, which is normally aversive for our rats. And just like you've seen before, we get a downward deflection in dopamine release during the intraoral infusion of this concentrated salt solution. But if we make the animals sodium deplete, give them the exact same stimulus, the exact same animals, the exact same stimulus, when they're sodium deplete, you see a very different dopamine response. Now there's a marked increase in dopamine during the intraoral infusion of sodium. One of the cool things about sodium appetite is it's very selective for substances that contain the sodium ion. So we did another experiment looking at a variety of different taste solutions: water, calcium chloride, lithium chloride, and sodium chloride. And under sodium replete conditions, when the animals have no need for sodium, no response of dopamine to any of them. Under sodium deplete conditions, we get a big increase to sodium, which you've seen before. We also get a big increase to lithium. Rats can't discriminate the taste of sodium and lithium. So when they get the intraoral infusions of lithium chloride, they register that as sodium, and you get a big increase in dopamine. But look at the response to calcium chloride, to water, no dopamine response whatsoever. So the response is very selective. It's really a taste by state interaction here. Taste combining with physiological state to drive that dopamine response to sodium or things that the animal thinks is sodium. What about satiety? You know, animals that are sodium deplete get access to sodium, they consume sodium, the lost sodium, and then they've had enough. And the solution presumably goes back to being aversive. So what I want to show you now, hopefully this will start over again. It's a GIF, and I'm still learning how to insert these things into my talk. No. It won't play again, sorry. But I think this is a really cool animation here, which I'll talk through. This is every trial when animals are getting that hypertonic sodium solution when they're sodium deplete. And what you can see grayed out are the initial trials where there's a big increase in dopamine when the animals need the sodium. Then over the course of that recording session, as the animals are regaining that sodium, now the solution is evoking ultimately a negative change in dopamine. And this blue line is just the average across the whole session. If you look at just the average across the whole session, you see that bump in dopamine. But what you don't see is the fact that it starts out quite high and then decreases as the animals are sated on the sodium solution. You can see that illustrated a bit better here. In light blue, you're seeing the decrease in the dopamine response to sodium in sodium depleted rats to a highly concentrated sodium chloride solution, 0.45 molar. Very steep decay in that dopamine response to the salt solution. Compare that to 0.1 molar in red, a much more shallow decay. And I think why I'm sharing this with you here today is that it illustrates that, again, physiological state, not just hunger, not just sodium hunger, but sodium satiety is somehow communicated to the mesolimbic dopamine system within a single recording session. And I want to explore that further with you in the next few slides. I showed you a few slides ago that there's no response to water in sodium deplete animals, but we can all relate to the fact, I'm going to take a sip of water because my throat is dry. We can all relate to the fact that we get thirsty and that water, when we're not thirsty, may be just kind of so-so. But if we've been hiking all day, we've lost a lot of body fluid, suddenly water becomes highly rewarding. And again, we can see that reflected in the dopamine response in our rats. Here are intraoral infusions of water when the animals are not thirsty, when they've had 24/7 access to water in light blue. However, when these same rats are thirsty, just overnight water restriction, now intraoral infusions of water evoke this very large increase in dopamine. Very basic stuff, but it allows us to then start asking, I think, important questions. How do the dopamine neurons come to know that the animal's thirsty? Here's an experiment where we paired a cue with the upcoming availability of access to water, in water-restricted animals, thirsty animals. Initially, on the very first day of testing, the animals don't know anything about the cue and its relationship to water. They're just thirsty. And eventually the water comes and they drink the water. What you're looking at on the left is the dopamine response evoked by the cue that ultimately comes to predict the water. No prediction yet. The animals haven't learned anything about the cue, so no dopamine response. And you can see by their behavior, they really don't know much about the cue behaviorally, in that there's a very long latency to when that water finally becomes available and when they go up and lick it. But by the very next day, by day 2, they start to get a hang of it. The cue predicts the availability of water. As soon as that water comes out, they're on it and they start drinking away. And if you look at the dopamine response, you can see it starts to emerge to the cue. We've known this for a few decades now, that dopamine responses emerge to the earliest predictor of reward, and you're seeing that here. Dopamine responses to the cue predictive of water in water-restricted animals. By day 3, they're very good at this, and you get a big dopamine response evoked by the cue. And just to continue with the idea that physiological state can modulate the signal, look at the difference here. Again, same animals when they're thirsty versus not thirsty. Dopamine evoked by the cue when they're not thirsty, small or non-existent dopamine response. However, to that same stimulus when the animals are thirsty, big dopamine response. Physiological state modulates, tunes the dopamine response to the same stimulus. Okay, so now we can ask, how do the dopamine neurons know that the animal's thirsty? Well, there's an area in the brain called the subcortical organ, which is kind of like a first responder to changes in body fluid homeostasis. And again, we've known this for quite a while. If you make the animals thirsty, or if you give them, mess with body fluid homeostasis by giving them an injection of hypertonic saline, making their increasing the osmotic challenge, or give the thirst hormone angiotensin 2 to the brain, that turns this region of the brain on, the subcortical organ. Here's just a quick example showing that in water-restricted animals, neurons of the subcortical organ become active. It's not just any old neuron, it's glutamatergic neurons of the subcortical organ seem to respond to these deviations in body fluid homeostasis. So what we did is we took control over these glutamatergic neurons of the subcortical organ. We expressed designer receptors exclusively activated by designer drug, DREAD receptors. These are normally inert. They don't do anything. But if you give the normally inert compound CNO, CNO will bind to these DREAD receptors, activate them, and those DREAD receptors will then modulate the excitability of the neuron. And so on the screen, you're seeing expression of the DREAD receptors in glutamatergic neurons. We can then give CNO and measure the neural response to activating those neurons. If we give vehicle, no change in their activity. However, if we give the normally inert CNO in cells that are expressed in that DREAD receptor, we clearly activate them. And this is just a quantification. The cool thing is we can also activate behavior. So just activating those glutamatergic neurons of the subcortical organ makes non-thirsty animals drink water. They are, by all appearances, they appear thirsty. They're not. They're in totally normal body fluid homeostasis. Just activating those glutamatergic neurons of the subcortical organ gets them drinking. And perhaps not surprisingly, based on what you've already seen here today, if we take non-thirsty animals that have had water 24/7, measure the dopamine response to a cue predictive of water, under normal circumstances, you see no dopamine response. Simply activating the subcortical organ by activating those DREAD receptors in these same animals, now we see a massive increase in dopamine activity, time locked to the cue, predictive of water. So I present this simply as an example of how physiological state might get communicated to the dopamine neurons. Here's a node in the brain, a first responder to a deviation in body fluid, somehow communicating with the mesolimbic system. We don't know exactly how that communication occurs, but knowing that it occurs, we can then, in our ongoing work, sort of work out whether this is a direct projection from the subcortical organ or more likely via intermediaries, and identify those intermediaries and understand how they're recruited to modulate the dopamine response. Okay, so I've shown you so far that physiological state can tune the dopamine response to the same stimulus. I showed that to you with thirst and water. I showed that to you with sodium appetite and concentrated sodium solutions. Now I finally get to come back to something that may be a little bit more of interest to a group that's focused on diabetes, and start asking about dopamine responses to food reward and how physiological state might modulate those dopamine responses. So again, another video. This time we have a hungry rat, and all we're going to do is just drop a sugar pellet in the cage 5 seconds into the video, and again, measure the dopamine response, this time in the nucleus accumbens. Okay, Hopefully what you saw is that the moment that the pellet drops, there's an increase in dopamine. It peaks at the moment that the animal retrieved the pellet and then falls back down to baseline just a second or two later. The fact that it's on the rise before the animal even gets its mouth on the sugar pellet speaks to this ability of cues that are predictive of reward evoking the dopamine response. The sound of the magazine turning, the sound of the pellet rattling down the chute is enough that the animals learn that the pellet's on its way and it can drive the dopamine response. But it peaks at the moment that the animal retrieves the pellet. And then we can again ask about physiological state. So the magnitude of the dopamine response depends on the physiological state of the animal. If the animal's under ad libitum fed conditions, modest spike in dopamine. If the animals are hungry, a significant potentiation in the magnitude of the dopamine response. And again, that gives us an opportunity to ask, I think, all kinds of interesting questions. What signals and circuits relate physiological state to the dopamine neurons? And there are many when it comes to food. We have been interested in a variety of them, including the quote unquote hunger signal ghrelin. Ghrelin is made and released by cells of the stomach, increases in circulation as we go further and further away from our last meal, acts in the central nervous system to stimulate feeding in animals. And so again, very simple experiment here, non-hungry animals that have had 24/7 access to food. We give them sugar pellets, they retrieve them, measure the dopamine response, modest spike in dopamine, and then halfway through the session, after about 10 sugar pellets, we just make an injection into the brain, into the lateral ventricle of vehicle. No physiological consequence, and you can see no change in the signal. Compare that to when halfway through the session we give ghrelin, and now we start to see something different, a potentiation in the magnitude of the dopamine spike when we infuse ghrelin midway through the session. Look at the dopamine spike when the animals are actually hungry. Relative to the vehicle condition just above, when animals are food restricted, there's a larger spike in dopamine. And in fact, it looks very similar to the response when the animal has had ghrelin injected. If we block ghrelin receptors in hungry animals, we see a decrement, a decrease in the magnitude of the dopamine response. So again, a little taste of data to support the idea that peripheral signals and circuits can relate physiological state to the dopamine neurons, either directly or indirectly, as we'll hopefully talk about at the end. And now I want to shift gears to other potential hunger signals, other potential signals that relate physiological state to the dopamine neurons, and talk just a little bit about cytoglucopenia. It turns out that if way back in the 1970s, that when you give a drug that blocks glucose utilization, like 2-deoxyglucose, that stimulates hunger in humans, and it also increases pleasantness ratings of sugar in humans. So it's a 1-2 punch. Humans think that they're hungry when they're not. And it also increases the rewarding value of sugar. 2-DG and a drug that we use, 5-TG, 5-thiodeglucose, block glucose utilization. You guys probably know this way better than I do, but the key here is that 5-TG potently stimulates feeding behavior even in sated animals. And so we wanted to know whether cytoglucopenia, and in particular, more specifically, whether 5-TG can modulate the dopamine response to sucrose as a means of communicating hunger to the dopamine neurons. So again, very simple experiment. We're just making intraoral infusions of a sucrose solution directly into the oral cavity of the rat. And here we're giving systemically different concentrations, different doses of 5-TG. And you can see that there's really no change in the magnitude of the dopamine spike here. This is injecting the animals 15 minutes before running the test. If we inject them 45 minutes before the test, Well, now you start to see a modest change, a modest increase in the dopamine response, suggesting that somehow the 5-TG has to diffuse away from the injection site to some other site to have its effects. Okay, let's give this stuff directly into the brain. Here we're giving it to the lateral ventricle. And sure enough, if we wait only 15 minutes, we get a potentiation of the dopamine spike. If we wait 45 minutes, we get an even larger potentiation of the dopamine spike. Again, suggesting that the critical site of 5-TG action might be remote to the lateral ventricle. Okay, inject it into the fourth ventricle, sort of directly accessing the brainstem. And now, again, 15 minutes in, we see a potentiation of the dopamine spike, and it's about as big as we're going to get. 45 minutes later, not much bigger. And if we just do a crude estimate of effect size here, doing a Cohen's d, you can see that really it's those fourth ventricle sites where we get our biggest effect. And this jives, I think, quite nicely with the literature. We know that hindbrain areas are critical for glucoprivic feeding. Again, relatively older literature. I'm getting more and more careful about what I call old, being old myself now. But if you inject 5-TG into the forebrain, but block its flow into the brainstem, you can block glucoprivic feeding. And finally, there's a little bit more recent data to suggest specific populations of neurons in the brainstem responding to cytoglucopenia that may project directly to the dopamine neurons. I think the take-home message that I want to leave you with here is that this is yet another mechanism for physiological state to be communicated to the dopamine neurons. I showed you already ghrelin, a peripheral signal that can act somewhere in the brain to toggle the dopamine spike. Here, cytoglucopenia, central cytoglucopenia, hindbrain cytoglucopenia can modulate the dopamine spike. One maybe unfortunate aspect of delivering 5-TG cytoglucopenia centrally is that it doesn't just induce hunger, doesn't just induce feeding, but it also causes a counter-regulatory response. There's a massive increase in blood glucose as a function of inducing cytoglucopenia in the central nervous system. These are primarily different pathways. The pathway by which cytoglucopenia induces feeding is an ascending pathway from the brainstem. It's a descending pathway for this counter-regulatory response with respect to blood glucose. But the bottom line is that we can't be sure that the dopamine response isn't somehow secondary to this counter-regulatory response. So we decided we would inject our animals with insulin, which also induces cytoglucopenia, but unlike 5-TG, causes the opposite response in blood glucose levels, a decrease in blood glucose levels. So we injected insulin systemically, and at first blush, again, very simple experiment. We're just making intraoral infusions of sucrose into the animal's mouth, measuring the dopamine response in the ventral tegmental area. And at first blush, it looks like insulin has no effect on dopamine signaling. But I've shared this with you with the sodium appetite data. It's really important to look at a trial by trial level. And when you do, there's a lot more than meets the eye when you just look at the session average. It seems as though insulin is indeed potentiating the dopamine response. It just happens to be occurring very early on in the session, and then the insulin response seems to wane over the course of the 30 trials that we give our animals. Okay, so let me review kind of the questions that I wanted to address with you today, and hopefully I provided some answers. So does the mesolimbic dopamine system differentially encode rewarding versus aversive? I
Just,
hope I
so
convinced you
Mitch,
the answer is yes.
so just want to
In our
mention,
hands, unequivocally.
just want to.
Oh, is there a
mention
question?
Yeah, this is Terry, calling in from New York. But
Hey,
just
Terry.
one associated thought before I forget it, okay? It might not come up later,
Yes.
is that,
Yes.
repeated insulin-induced hypoglycemia causes hypoglycemic unawareness. So there is, central adaptations that might be occurring to limit, this response later on. And just wanted to throw that out there.
Yeah, I thank you for that very much. And I am, as a father of a type 1 diabetic, I'm very well aware of that phenomenon. But not just as a parent, I'm hoping to work with Vanessa Routh in New Jersey, who is interested in sort of hypoglycemic, I'm sorry, you said it better, unawareness or
Yes.
yeah, hypoglycemia unawareness. And she has a hypothesis about lateral hypothalamic orexin neurons, which we know project directly to dopamine neurons. So yeah, I thank you for the comment. I'm very much aware of it. And this is why I think it's fun to talk to this group in particular to sort of highlight these changing signals that I may not know as much about, but impact the signals that I do know a lot about or care about.
Yeah, thank you.
Sure. So again, we can circle back to that point, but let me just run through the questions I want to address. Reward versus aversion, in our hands, 100% yes. But I want to make sure that I am responsible about my unequivocal statement in that we've only measured from select areas of the midbrain, the paranigral region of the ventral tegmental area, and only one of its targets, the lateral shell of the nucleus accumbens. So it remains possible that different dopamine populations, different dopamine terminal projections might encode reward versus aversion in a different way than I've shown you here today. Does the mesolimbic dopamine system flexibly encode the same taste stimulus as its value changes? Again, I hope I convince you that the answer is yes. I showed that to you with conditioned taste aversion and with sodium appetite. But we still have a lot of work to do in this domain. We want to identify the critical inputs and their plasticity to understand how the dopamine system, how the dopamine signal can be flexible based on learning or motivational state. Potential sources that we are currently interested in are the amygdala and the bed nucleus of the stria terminalis and the lateral hypothalamic area, which I've previously mentioned. Finally, how do dopamine neurons come to know about the physiological state of the animal? Well, there are lots of questions that that might provide possible answers. Circulating factors for body fluid homeostasis, angiotensin 2, for hunger, ghrelin, for satiety, GLP-1. Is GLP-1 released by the intestines acting in a paracrine fashion to modulate the vagal input to the brainstem? Is GLP-1 being released centrally to tune dopamine signaling? I didn't get a chance to show you our GLP-1 work, but we've shown that GLP-1 agonists can suppress dopamine signaling to food reward. How are the first responders, as I call them, communicating to the dopamine system? The arcuate nucleus, which many of us think of as a first responder for caloric deficit. The HRP neurons, in particular of the arcuate nucleus, there's some data to support the idea that they can project directly to the VTA, but they project to a lot of different places. And so is the arcuate nucleus involved, and how is it involved? We're interested in brainstem targets, the nucleus of the solitary tract. And finally, circuits within the ventral tegmental area itself, the nociceptin neurons seem to increase their activity as animals reach satiety. And so maybe that's a node that is communicating or toggling or modulating the dopamine spike to food reward. I still wrestle with this final question up above. And in 2018, a colleague of mine published a review paper that titled, "What Does Dopamine Mean?" And I added even up there. What does dopamine even mean? We're still, believe it or not, trying to figure that out. And I hope some of my data point to the meaning of dopamine. Decades ago, we thought we knew everything and that dopamine signaled a reward prediction error. Others posited that dopamine was important in the psychological phenomenon of wanting or effort needed to acquire a reward. But more recently, some folks have put forward some interesting ideas that dopamine is signaling perceived salience. I suppose the most current idea that I like the best, that I'm coalescing around, is that dopamine is critical for the formation of learned actions that lead to reward. And believe it or not, I think that gets us closer and closer to the original hypothesis of what dopamine was doing that was put forward in 1978, and what everything old is new again in some ways. But I just thought I'd leave you with this idea. Again, I don't know how often you hear about central circuits. I don't know how often you hear about central motivational circuitry, but we're still wrestling with exactly what dopamine might be doing in the brain. And I just wanted to acknowledge that unsettled situation. Ultimately, I think it's enhancing the probability of approach and consumption. And I think that really makes it an interesting target for the treatment of disorders of motivation like overeating and obesity. And could physiological state, could hunger be a risk factor for food seeking through enhanced phasic dopamine signaling? And whether homeostatic signals like ghrelin receptors, like GLP-1 receptors, could they be good therapeutic targets for the treatment of disordered motivation, like overeating, drug taking, through action on dopamine neurons? And so with that, I want to thank the folks that did the work. I just sit in my office these days, but really talented people are doing the work. With respect to the conditioned taste aversion work, that was mostly acquired by Samantha Herr, but Max Lowe is ramping up her activities, and Rachel Donka gave us a lot of help with some of the analyses that I showed here today. The sodium appetite work was, a lot of it was done by folks who have moved on from the lab, but the current work with the slopes that you saw there, that's work being done by Paola Bazzino here in the audience today. The work on cytoglucopenia, 5-PG and insulin was all done by graduate student in my lab, Vaibhav Kananur, who sadly for me is graduating in a week, hopefully. And it's been a lot of fun having him. So I just want to thank those folks and many others. Thank you for your time, and I'm happy to take any questions or comments.
I'll just go ahead. I think, really interesting